Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma, and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is the cover article from our recent summer 2019 issue, titled Automating Automation: How Close Are We to Artificial Intelligence Impact? This article is written by Dr. Michael A. Tarselli, Dr. Yohann Potier, and Dr. Alan E. Fletcher. So now on to the main article. Automating Automation: How Close Are We to Artificial Intelligence Impact? In terms of consistency, repeatability, known errors, and sheer volume, there exists perhaps no better collection of data for computer learning than that emerging from automated processes. Many common lab procedures now run in parallel miniaturised experiments: DNA synthesis, target screening, organoid culture, genetic analysis, organic reactions, safety assays, which are poised for extensive curation and algorithm development over the next 10 years. This article briefly outlines each area and offers opinions about how close we are to having artificial intelligence, AI, deep learning, DL, or machine learning, ML, influence each scientific domain. The past 10 years have seen an amazing change in the miniaturization, cost reduction, high fidelity, and data acquisition of modern instrumentation. The surge of robotic control processes enabling DNA synthesis, genome editing, screening, plating, and cell culture have led to a data explosion. In the 90s and early 2000s, the introduction of automation and high-throughput screening transformed the way in which drug discovery research was performed, leading to a rise in the number of compounds tested against the target of interest and a significant amount of investment in the quest to produce the ultimate screening factory. Massive repetition led to consistency, lower error, greater precision with higher number of experiments, better fidelity, and the ability to quickly generate enough data to run in silico or virtual experiments. This boon has generated yet another problem, that of big data, where sophisticated algorithms must infer patterns from large warehouses of data to distill wisdom from gathered information. Now, as many researchers struggle with the ever-increasing complexity of drug development and the rise of personalized medicine approaches, the increasing use of artificial intelligence and deep learning presents one of the most promising and transformative opportunities for the life sciences and medical industries. The emergence of artificial intelligence and deep learning in drug discovery provides many advances over traditional techniques in genomics, image analysis, and medical diagnostics, and is one of the reasons that pharmaceutical companies such as Merck, Sanofi, AstraZeneca, and Takeda are placing big bets on the ability of AI to deliver improvements in quality, clinical success rates, and reduced costs. This article will show how recent revolutions, chemical and biological automation, produce enough data and learning to build a deep learning model pipeline, making science faster, more efficient, and more accurate. Chemistry automation: computation, synthesis, and prediction. Prognosis: available and maturing. As a discipline, chemistry has existed for thousands of years, and its practice on industrial scales in recent history dates back at least a few centuries. As such, AI and DL and machine learning can more readily be applied here. Multiple large datasets exist in the public domain, albeit of varying quality and reproducibility, and all large companies have proprietary molecular databases containing molecular descriptors, predicted properties, safety thresholds, and assay data against multiple targets of interest. Most applications in artificial intelligence and the subset field of machine learning in chemistry are concerned with predicting three things: which molecules to make, how to make them, and predicting properties or safety panel data for the molecules thus synthesized. To be clear, applying algorithmic intelligence to synthetic pathways is not new. Nobelist E.J. Corey explored symbolic logic and computational route finding with his LHASA and OCSS already in the late 1960s. Chemistry AI generally, and machine learning from text mining specifically, has been used to great effect. One of the authors of this article, assisted a Novartis team with a text mining and predictive reaction assignment of 30 years of patent literature, seeking to describe en masse the types of molecules and physical properties produced by pharmaceutical and biotech firms. Retrosynthetic analysis now commonly runs on multiple commercial platforms, such as SciFinder, formerly ChemPlanner, or Millipore Sigma's Cynthia, formerly Chematica. Both systems utilize machine learning to predict routes based on combinations of expert rule sets and literature-derived reaction capture. Multiple companies have begun to use fully AI-based approaches to target selection, drug design, property prediction, or synthetic execution. Cyclofluidic utilized AI-based algorithms to analyze product output to inform future synthesis rounds, an approach adapted and refined by ex-Scientia. Nimbus Therapeutics claims to utilize bespoke computational tools to shorten drug discovery development times by 75%. Revolution Medicines, which spun out from synthetic automation efforts at the University of Illinois, uses a proprietary computational pipeline against oncology targets, while Recursion Pharmaceuticals mines phenotypic data to construct models for ideal drugs. Though not itself AI, data collection and automation efforts on an academic scale, fully digital synthetic engines have sprung up in labs such as Lee Cronin's at the University of Glasgow. One risk for chemistry is that in many small firms and academic labs, data is still recorded in paper lab notebooks, which slows the amount of AI adoption to early discovery and more risky reactions. Automation provides consistent data templates and inputs for algorithms, which means a gap to overcome would be to decide which processes are automatable. Perhaps, due to the large data warehouses and mandated electronic storage of experimental records and instrument data, practical innovations in this space from industry have outpaced academia. Another cautionary tale has come from the use of sophisticated AI systems such as IBM's Watson, famous previously for accomplishments in beating human competitors in Jeopardy and assisting doctors with medical literature gathering. However, IBM announced in early 2019 that it had halted drug discovery applications of its system. This may also be an indication of the maturity of AI in this field. IBM's exit was not seen as an algorithmic issue, but rather one of insufficient high-quality data on which to base drug predictions. To be certain, as observed in Ian Davies' seminal Nature perspective, we face many challenges to wider adoption of AI in chemistry automation. Consistent data formats, standard automation sets, cultural shifts in reporting and practice. Discovery biology, from DNA to screening. Prognosis, gaining traction. Various methods of AI have been successfully applied in drug discovery outside the small molecule world. AI, and more specifically deep learning, are useful on several levels for life science and biologists in particular. AI improves the quality of the obtained insight and allows scientists to handle the large volume and speed of data coming from automation that would otherwise require superhuman effort from a single bench scientist. Biological deep learning allows scaling up of effort and expedites traditionally manual steps. These successes depend heavily on several factors: the amount of data, the type of data to analyze, and the methods used for the analysis. Unlike most bench chemistry, diversity in biology data is the main hurdle. Another obstacle, machine learning, requires large amounts of data across the range of expected measurements of a given experiment, yet also a large amount of data necessary for the training, validation, testing, and finally execution of the machine learning method. Though a general biology agent does not yet exist, deep learning methods have been applied to more narrow questions with success. One application, phenotypic screening. Just like image recognition of cats, we can leverage similar algorithms for microscopy image analysis. Image-based screening is particularly suitable for high-throughput cell biology, where microscopy images provide scientists with means to determine different types of cells in a sample, counting the cell or if proteins are expressed, or to identify cell anatomy. One deep learning model, the convolutional neural network, CNN, was developed for image processing and has been applied to microscopy image analysis. Other methods of deep learning have been used in different contexts for image analysis. Histopathology images can be analysed to determine a phenotype associated with gene expression. Such work would have previously required manual inspection of the slides. Finally, morphological classifiers of cell phenotypes upon small molecule exposure have recently been analysed with CNNs. Another successful area of application for deep learning is genomics. Large amounts of data in the field allow leverage of machine learning models to identify biomarkers associated with phenotype or level of gene expression. The amount of data generated from whole genome sequencing is massive, and collaborations between academic and tech firms to capitalize on their analysis have increased rapidly. For example, in that between Google and the Broad Institute to implement the Broad's open-source genomics toolkit on Google's cloud servers. The same methods can be used to search for therapeutics with the potential to silence genes or inhibit specific gene activity. For example, artificial neural networks, ANN, have been used to predict siRNA activity on predefined target sequences. Another interesting deep learning application covers the design of new peptides with high bioactivity. Combinatorial peptide libraries challenge creators through the sheer numbers of generated compounds. It is then crucial to select the peptides with the highest activity for synthesis. Machine learning provided a method to predict that activity and sort the selected peptides. Other machine learning methods provided a way to predict protein binding site for small molecules, or prediction of protein expression and solubility of a large dataset to optimize high-throughput experiments. These projects would not have been possible without the use of both automation and machine learning. Specific models and methods for machine learning are keys to answer specific scientific questions. The diversity of methods is key to the diversity of biological questions. It is why the industry has been turning to technology companies for models such as GAN, generative adversarial networks, for customized hardware, or for collaboration with experts in machine learning algorithms, coupled with large-scale cloud capabilities. Entire companies' business models are based on machine learning methods for drug discovery. The unique combination of cloud technology, automation, and machine learning provides opportunities in life science at a scale we could not manage before. Instrumentation, image analysis, IoT, virtual help. Prognosis, imaging and voice interaction prototypes, wider adoption pending. The concept of a robotic scientist is not new and was first conceptualized in 2004 as a combination of computational methods, automated instruments linked to complex laboratory robotic systems, and the need for AI and machine learning to test and iterate on a hypothesis in real time. Cellular imaging provides an ideal opportunity to showcase the power of AI. High-content screening, HCS, or cellular imaging and analysis is today widely used across many parts of the drug discovery process, driven by the need to gain greater understanding of the phenotypic nature of interactions between potential new drug candidates and the cells found in the human body. The ability to predict and visualize potential unwanted cellular interactions increases the potential of success when a candidate enters the clinical phase of testing. However, the vast quantity of information provided by even the most basic of HCS platforms opens itself to the need for more intelligent forms of data analysis. AI techniques provide the ability to provide enhanced segmentation and even to identify specific organelle structures in the absence of segmentation, precluding the need for cell labeling. In addition, so-called image-based cell profiling strategies provide a path to high-throughput quantitation of phenotypic differences in cell populations through the collection and analysis of hundreds of morphological changes caused by prior treatment with a chemical or biological agent. This analysis approach provides a path to identification of novel targets or mechanisms of actions for prospective new drugs. Given the number of sensors and facile communication across various lab instrumentation, a growing movement seeks to capitalize in two distinct but complementary ways: building Internet of Things networks to alert scientists, or two-way communication using robotic lab assistants powered by natural language processing and virtual models. In summary, automation itself has provided the high-quality, high-volume data required to train neural networks and recognize patterns. As new tasks are automated for both chemistry and biological processes, the instruments also gain sensor data: location, position, temperature, torque. Soon, it will not be even that a gene is properly functioning or a reaction works, but the more subtle ways of discerning how the robot or instrumentation runs the assay that will be the rate-limiting step to scientific innovation. A note from the authors. This article was commissioned in part to celebrate the inaugural AI in Process Automation Symposium, held in Boston, Massachusetts in October 2019. The Society for Laboratory Automation and Screening, SLAS, of which all authors are volunteer leaders or staff, appreciates DDW's kind invitation and facilitation to produce this piece. This article was written by Dr. Michael A. Tarselli, Dr. Yohann Potier, and Dr. Alan E. Fletcher. Dr. Michael Tarselli trained as a synthetic organic chemist and worked in CROs, startups, and multiple pharma companies as a chemist before leading a team in chemical information systems development at Novartis. Since 2018, Mike has served as scientific director for SLAS. Dr. Yohann Potier trained as a computational chemist and spent time in business analysis and informatics before landing at Novartis. He leads a team creating new solutions to support bench scientists across the biological and bioinformatics community. Yohann is also involved in local consortia such as Potier Alliance. Dr. Alan Fletcher trained as a pharmacologist, then spent a decade running HDS and robotics for MSD in the United Kingdom. On the business side, he has served as a director and VP GM for life sciences at 2 technology firms. He is the 2019 president of the SLAS Board of Directors. If you've enjoyed this episode, then you can subscribe to Drug Discovery World free of charge by visiting our website at ddw-online.com, where you can also view all of our articles including references and images, and download the original PDFs. The links are in the show notes. If you've enjoyed the podcast, then do leave us a review and subscribe, and you can also follow us on LinkedIn and Twitter. Thank you for listening, and we'll hope to see you in our next episode.